Hello everyone, my name is Omchit Surien from Universiti Kebagsaan Malaysia. So today I'm going to share my research work entitled Histopathological Analysis of Pterostilbene as Chemopreventive Agent Against Lung Squamous Cell Carcinoma in Mouse Model. So for the introduction, lung cancer is divided into few types based on the histopathological observation. So in this study, we are focusing specifically on squamous cell carcinoma, which is one of the common type of human lung cancer. So the lung SCC or lung squamous cell carcinoma is defined as the abnormal growth of the cell from the bronchial epithelial cell to the hyperplasia metaplasia, and lastly we progress to become a cancer or SCC. So other than histopathological observation, lung SCC also can be differentiated from other type of lung cancer through the expression of cytokeratin 5-6, where usually lung SCC tumor has the overexpression or highly expressed cytokeratin 5-6. According to the latest statistic, lung cancer is one of the common type of human cancer, and lung cancer is leading cause of cancer-related death, where the high mortality rate among lung cancer can be contributed by many factors, and one of them due to the late diagnosis, where most of the lung cancer patients were diagnosed at the advanced stages. This late diagnosis will increase the risk of treatment failure that lead to the high mortality. So the chemoprevention is one of the alternatives to the treatment to reduce the mortality rate, and the chemoprevention is defined as the use of any natural, synthetic or biological agent to delay, suppress or reverse the carcinogenesis. So in this study, we investigated the chemopreventive effect of pterostilbene against lung SCC. So this pterostilbene is a natural compound that can be found in a grape, blueberry and also tree wood of Tecopus marsupium. This pterostilbene is a potential to be developed as a chemopreventive agent because of pterostilbene has been proven to exhibit the various pharmacological properties such as anti-inflammatory, antioxidant, anti-proliferation and also anti-angiogenesis and metastasis. Moreover, this pterostilbene also has been proven to exhibit the anti-cancer against lung SCC cell line in vitro and also in vivo mouse xenograft model. However, the chemopreventive effect of pterostilbene against lung SCC in chemically induced mouse model has yet to be determined. So this is my research question and my research hypothesis. So for the general objective, to investigate the chemopreventive effect of pterostilbene against lung SCC in mouse model. And the specific objective, my first specific objective to determine the effect of pterostilbene on lung SCC through the histopathological observation and also thickness of bronchial epithelium layer. And my second specific objective to determine the effect of pterostilbene on semi-quantitative histopathological scoring. And my third and also last specific objective to determine the effect of pterostilbene on the protein expression level of cytokeratin 5-6. So next, I move on the methodology. So in this study, the total of 24 female biopsy mice were randomly divided into 4 groups. So for the first group is the vehicle control group, where this group treated with the corn oil through the intraperitoneal injection that act as a vehicle for the pterostilbene, and 70% of acetone that act as a vehicle for the carcinogen through topically at the shaved subscapular area. And the second group is the cancer group, where this group is treated with the carcinogen and TCU to induce the lung SCC. And the third group is the low dose of pterostilbene, where this group treated with the 10 mg per kg of pterostilbene together with the carcinogen. And the last group is the high dose of pterostilbene with the 50 mg per kg of pterostilbene together with the carcinogen. So after 26 weeks of the treatment with the twice a week, all mice were sacrificed and the lung were collected for the test. So for the objective 1, we proceed with the H&E staining, hematoxylin and eosin staining. For the histopathological observation under the light microscope. And for the H&E staining also, we measure the thickness of epithelium layer using the ImageGFG software. And for the histopathological semi-quantitative scoring is based on the pre-malignant stages in the development of lung SCC. And for the objective 3, immunohistochemistry was used to investigate the expression level of cytokeratin 5-6. And the quantification of cytokeratin 5-6 also was done using the ImageGFG software. So next, I move on the result and discussion. So for the first part is the histopathological observation. So as we can see here, the vehicle control group show a normal characteristic of lung histology, without the thickening of the bronchial epithelium layer, and the high magnification is show the normal characteristic of bronchial epithelium layer with a single ciliated columnar epithelium and basally located nucleus. In contrast, the group that received the carcinogen without psilocybin show the thickening of the bronchial epithelium layer, and at high magnification it show the disorganization of the cell with the increased nucleus cytoplasm ratio and also the formation of keratin pearl. And the low dose of psilocybin only developed the hyperplasia at the bronchial epithelium layer, and the high dose of psilocybin still maintain the normal characteristic of bronchial epithelium layer with a single ciliated columnar epithelium that's similar to the vehicle control group. Next, we look at the histopathological scoring. So as we can see here, the cancer group score the highest, with the mice in this group usually develop dysplasia and SCC in the lung. And the psilocybin reduced the histopathological scoring, with the most of the mice develop the hyperplasia and still maintain the normal bronchial epithelium layer. So next, we look at the thickness of bronchial epithelium layer. The cancer group or NTCU significantly increased the thickness of bronchial epithelium layer, but psilocybin treatment significantly reduced the thickness of bronchial epithelium layer. And this increasing in the thickness of epithelium layer related to the severity of pre-malignant stages in development of SCC. Lastly, we look at the protein of expression level of the cytokeratin 5/6. So we can see here the cancer group significantly increased the expression of cytokeratin 5/6, but psilocybin significantly reduced the cytokeratin expression level of cytokeratin 5/6. So from our finding, we conclude that psilocybin was able to prevent the development of lung SCC in mouse model. Therefore, the further investigation is needed to understand the underlying molecular mechanism of psilocybin as a chemopreventive agent. So this is my references. Acknowledgement. Thank you.